So Vivek is a computational biology senior research scientist in bioinformatics and data mining department in Novo Nordisk in Denmark. His work involves in silico biomarker and target discovery in chronic disease setting by integration of big data from bulk, single-cell, multi-omics, along with relevant phenotypic measurements, leveraging the interdisciplinary knowledge from biology, computer science, mathematics, and statistics. He also explores the application of advanced statistical machine learning and deep learning models in high-dimensional single-cell multi-omics data for particular problem scopes given the data bandwidth as part of some of his curiosity-driven research activities. So today he's going to talk about the role of bioinformatics in present era of high-throughput biology and drug discovery. At this point, can I just remind everybody, if you have any questions for any of the speakers, please add those into the chat box, into the question box, and we will be addressing these in the round table after this final talk.
Thank you very much, and really happy to be here. Thanks for the kind introduction, and thanks for the great panel speaker. So I will take a bit of a different dive into what all the other noted speakers have done. I'll basically speak about the role of bioinformatics in the present era of high-throughput biology and drug discovery. And before going deeper dive, I'll just give a kind of what I do. So I'm a bioinformatician, and there are different ways of how bioinformaticians are regarded or seen as in different parts of the society, or let's say scientific society. But what I basically do is I have a problem scope. We get a lot of different sequencing data from different modalities, and then I actually take those data, analyze them, use different kind of models, and then try to discover genes or proteins that can help us in terms of in silico drug discovery, which can fall into these four corners of the medicines. So prediction, prevention, participation, etc. Now, in Novo Nordisk, we have a very, we're trying to build more and more human-centric approach, where I belong to the Bioinformatics and Data Mining Department, which is a part of the AI and Digital Research big department. And we cater to different kinds of, areas like we have different therapy areas that we cater to for different kind of service projects and research projects in part of the drug discovery pipeline. And then there are different departments like research technologies, the translational medicine, and then role of bioinformatics is now also catching up in the clinical trials, where you have a certain kind of mechanistic trials, which we are using single cell technology to understand the mechanism of actions of certain drugs, so that we can improve in future as well. And the kind of different kinds of work packages or kind of things that we do here as a part of this is we support early drug target identification, builds analytical workflows for target evaluation, biomarker identification, and data visualization. We use different kinds of data sets that are public or internal omics data that can be around, it ranges from different types of omics, bulk, transcriptomics, proteomics, metabolomics, and genetics, a bit of epigenomics. We're exploring that part as well. A lot of single cell omics these days, and mostly we start with the human data, and then we go to the in vitro and in vivo settings to derive biological insights. So my outline of the talk will be divided into mostly three parts. So what is bioinformatics in time of high throughput biology? Why is bioinformatics important in drug discovery? And the future state and aspirations of modern medicine. So the first one, which is why is bioinformatics what is bioinformatics in terms of high throughput biology? So modern medicine and biology is going a kind of revolution. In the last 10-15 years, we've realized that now we are actually in an unprecedented era where we can actually leverage the power of not only the diagnostic information that we had in the medical field or biological field, we can actually couple them with the novel, high throughput information that can be generated via sequencing technologies, and that can profile different informations, be it the genetic blueprint, or be it the regulatory informations that how genes are expressed or regulated in terms of transcriptomics, or the more downstream translational information like that of the protein information, the lipid and the metabolites. And these can be done also in terms of different organs, in terms of the cellular, and we can derive this relationship in terms of multiple networks. And thus, if we can actually reach a much more a big high throughput information complex where we can actually understand how the disease progression or the trajectory has happened from starting from the healthy to the disease, or even different stages of the disease using these kind of sequencing technology. And what we try to achieve is basically to understand the central dogma of the molecular biology, which I think is the hardest part of the science. And we can actually understand through these kind of information, where has the insult happened and what is driving these insults, and how that is actually reaching to the disease counterpart, and what gives us a disease phenotype. And in the end, what it's trying to achieve is basically a genotype to phenotype information that can help us to get better understanding of the disease biology as well, and then eventually lead us to early drug, in silico early drug targets, which can then feed into our drug discovery pipeline downstream. And this is where bioinformatics come into picture, because all these things are omics, and this is where a bioinformatician or data scientist all come together to analyze and help build predictive models to reach these kind of fruition. Now, why is bioinformatics important in drug discovery? Well, medicine for the last few years is undergoing a paradigm shift. We know that the one-size-fits-all theory is not actually great. Not every medicine works in every patient. There are adverse events, there's no benefit, there's some benefit. And we have moved, and we have been moving actually from one size to stratified medicines, where patients were grouped into different disease subtypes, risk profiles. And then with the era of the Human Genome Projects and more omics, we are now doing something called precision medicines, where we can actually, we're aiming to actually get much more precision-based and personalized-based, approaches to target disease, to find, tailored population where you can actually help them to benefit with the drugs. However, over the years, we've also learned that the drug development journey is actually very complex. It's very slow and expensive. Most of the candidates fail. Not all new drugs have great effects. Some are marginal. And this poses a problem, right? As our other noted speakers have already alluded to most of these things. And that's why we need some more effective and clever solutions. So what I do is, or I try to understand, can these solutions be brought into using different kinds of omics data? And if you marry these omics data that can be coming from getting an information of your DNA or your epigenetic information, that is the proteins that are binding around the DNA but not directly having a mutation on the DNA, but they're regulating the transcription of the translational machinery, or transcriptomics, which can also help with disease mechanisms, and proteomics. If we can take all this data together, and even actually we can integrate them with clinical measurements or health records information from those patients, along with some imaging, and come up with certain statistical and machine learning tools, we can actually help get candidate biomarkers in the in silico driven manner. And this is how the era of omics driven medicine works, right? And there are multiple applications of it. Some are like population genetics and genomics, where you do the GWAS, understand the polygenic risk scores, developmental biology, where you can, or disease biology, where you can do multi-omics of different organs and tissues and cells. Then you can also do multi-omics in terms of longitudinal study profiling, and all these can feed into early drug discovery and therapeutics. These are the kind of different departments that we have, where we are using the power of bioinformatics, and that I've listed, and the ones which are highlighted are the different kind of projects that I'm involved in, and the kind of therapy areas I have been working on a lot in the last 4 years of my involvement with Novo. However, there is an issue. These kind of things lacked cellular level information often, because we initially in the last 4 or 5 years, before the last 4 or 5 years, we did not have a lot of information about the cell types. And now and we know that human body has over trillions of cells. Some share commonalities, some don't. And this is where the field moved into something called single cell, and the era of the single cell popped up, and it has been tremendously powerful in terms of what it can do. And we are just scratching the surface, because it's very early on where you're realizing what it can bring in value, and both at the early and also in the late stage of the research. And there are different kinds of single cell profiling that can be done presently, which can actually complement and boost our entire early drug discovery pipeline. One of the recent example, what I would like to put in front of everyone, is the Human Lung Cell Atlas, which was a very big joint effort, I think, across Europe and US, as a part of the HCA, the Human Cell Atlas initiative, which tried to actually, profile around 2.2 million cells, or for 400 individuals, to actually understand or annotate the different cell types that we have in the human body, in the human lung, and what are the effects of the age, sex, and different kind of measurements that can affect the cell type and the underlying genes that express those cells, and if there is a SNP. And it is also trying to understand if there are kind of new cell types, if there are transitional states that happens over the course of the disease progression. So these are really, really very interesting findings, where you can actually basically find a Google map of the entire lung. And I think this also builds up a huge information that can help us with a lot in terms of when we think of early drug discovery, where we have an understanding of the gene, its behavior over the course of the disease trajectory, and which particular cell type it is present and how it is changing its course. Now, all said and done, this is great, but this is also a very complex thing where we say that we want to find a very specific single target gene or a single target protein that we want to push forward as a drug discovery pipeline. Because we often end up with a huge list of genes or a huge list of proteins that that can be a kind of needle in the finding needle in the haystack kind of chase, because we have patients' data that can come from tissues, biofluids, for which we have done omics or non-omics kind of information, like having phenotype imaging, and then omics. We can put them and parse them in different kinds of activities that can be, patient stratification, biomarker discovery, disease classification, drug repurposing, and drug discovery using different kinds of statistical methods, or even, advanced statistical methods, traditional statistical methods, and more advanced machine learning or AI based on what the question is and what kind of data sets we have, so that we can reach in terms of the personalized medicine. However, we often end up with big lists, right? As you can see on the left, these lists can range between 50 to 100, where we know the effect sizes, what is the uncertainty level of these genes that associates with the disease progression, and how they correlate with the clinical markers of interest or the FD endpoints, right? And this is where I think I take refuge of the knowledge graph scientists locally, or the publicly available knowledge graph databases, where I will often try to prune that list to in a smaller subsets, where I try to understand how well the genes that I have or the proteins that I have found, what are the functional representations of the categories, whether these genes really associate with the anatomy, or there is a disease gene relationship, or if there is a compound associated with it which can be perturbing its effect. And these all feeds into different kinds of applications that can be, based on multi-omic relationships, where for a particular gene, if you have an understanding around the DNA or around the transcriptome or the proteome or the interactions, it can help us with any kind of pharmaceutical applications like drug-drug interactions, side effects, and drug repurposing, and then any kind of clinical applications. So this is where I think the knowledge graph application comes into picture. And again, I am not a knowledge graph scientist, so I am looking at more on the end user as more of an applied form. And I've been, very grateful for the scientists who have actually put forward these kind of amazing algorithms that are out there, and even tools that I've been using at times for certain questions that I do address at my end when I end up with a large list of genes. And this is where I use them. And what I try to figure out is, now if I have these kind of big, lists from the omics data, and can I come to a very small subset of, targets? But having that means I have to really understand what are the different pathways and the networks that can be enriched from these genes. And for that, I need a knowledge base which are well curated, where you have the different kind of informations, where the relationships are very well derived. And this is where different kinds of knowledge graphs can help us, which can, to an extent, help us to derive the mechanistic hypothesis, do a causal reasoning, give us some candidate targets based on different kinds of network theory or neural network-based or graph network-based methods, which eventually will help us prioritize certain targets based on the community detections or centrality, whatever information that we can get, and help us decide from these lists if we have some that associates with the disease, some associated with the toxicity, and then we can identify some small group of very solid high confidence targets with degree of confidence. that can help us understand the disease mechanism, which can then go through to any kind of heat and lead identification downstream for any kind of drug mode of action. And this is how I have been thinking around and using it for the purposes of the work and the problem scopes I have been addressing. But this also means that we also end up with a huge high-throughput biological data information that often leads to a big, multimodal, high-dimensional complex data based on how we define it and the number of features that we have, the number of observations we look into, the overall dimensions it yields. That's because of the features that comes from different places and what kind of samples we look into, be it from patients, animals. It becomes a problem it becomes a mathematical problem or a data science problem, whatever you might say it fit into. But it does fit into the problems, as you can see here, like the curse of dimensionality, overfitting, the linear versus nonlinearity issue of the structure. And then again, once we fit all these things models, how much generalizable our output can be. And this is where I often feel that, and this is a slide I often reuse at my end, I always take a step back that it's all great what we're doing, but we have to also sometimes think back that what are the kinds of methods and the models we're using, whether they're really doing well in the current problem scope that we're trying to solve. And this is where different kinds of statistical measures or even advanced machine and deep learning models come into picture. Interdisciplinary bioinformatics is also very challenging, and I have written about it in terms of research purposes of how we can think of the multi-omics field. And then when we talk about high-dimensional data in terms of single cell, there's also a lot of problems, as you can see here. And then now the constructing of knowledge graphs and their applications, they're huge. They have a huge potential. But then there are a lot of problems as well when we think the data sparsity issue, the heterogeneity, the harmonization, and flexible analytical models and frameworks. And we can take care of these things if we are able to do systematic benchmarking efforts and then deploy these things in a proper way when we do more scalable research that we want to extend across different therapy areas. And this often becomes a very big challenge when we think of drug discovery. At least in my experience, when I see how the fields are moving, because we have tremendous potential, but we still have our complexities. And I'll take a bit of a step back. In the last few slides, I will just talk about certain things that I often do as a curiosity, different problem scopes and solutions. I'm very much interested in method building and model building. So I often try to get curious students with whom I can work for building bulk multi-omics integration methods. Sometimes I also focus on single cell field where I'm trying to understand what are the different dimension reduction methods or cell time annotation methods are there that we can improve or we can tweak. So I will focus on two problems that I will discuss that we have recently published and got accepted. One is how to better integrate multi-omics data by improving the feature extraction issue, and can we improve a single cell annotation using better embedding. So this project was a very curiosity-driven project with a student back in India, where we tried to integrate multi-omics data by improving the feature extraction problem. We are basically predicted in using the RNA or the gene expression using the DNA methylation and copy number variation and the protein coding regions. And we used basically a deep denoising autoencoder and a multi-layer perceptron to do this task. And then we tried to basically benchmark this against the different state-of-the-art machine learning algorithms, because we also wanted to understand whether our model was really something that is better than most of these fascinating methods that are already applicable. Then otherwise, why will somebody use our methods, right? And we found that the different variants of a model had really different performance, and one of them was actually on the top when compared to all the other models when we did different kinds of hyperparameter regularization. Finally, there's another problem that I'm very much interested, because I really enjoy high-dimensional data, multidimensional data, and the power of such can be really seen a lot in the single cell field. And then single cell analysis typical can be divided into two parts. One is at cell level, and the other is kind of gene level. On the cell level, there can be different kinds of analysis, like cluster analysis or trajectory analysis, which can help to understand the disease progression of the transcriptional states that reaches the disease stage. I am particularly interested interested in clustering and cluster annotation, so cell type annotation. And recently, we developed a method called Castle, which is a cell type annotation method for use for single-cell transcriptome data using semi-supervised learning. This has been accepted 2 months back. Unfortunately, it's still not online yet. Not sure why. But what we try to think is if this is a problem of cell embedding. So we basically came up with a semi-supervised method where you have limited number of labels, but are they able to actually, still predict your cell types for particular single cell data sets? And we thought it most on a problem scope of a cell embedding and a clustering issue. And we then studied it on different data sets that are publicly available to understand its performance and accuracy. And we found it was pretty strong and high. And when we compared it with different state-of-the-art methods like that of a semi-supervised, similar kind of semi-supervised method, which is built on sparse logistic regression and graph level ablation, then with unsupervised Seurat, and certain supervised methods, we actually found out that our model was actually working well in most of these data sets, and they were having a very nice performance. And this is how we have been actually able to see what's the contribution of our method. And finally, my ending slides will be future aspirations of modern medicine. Well, we are really moving to a kind of very big data kind of revolution in the medicine field. I think everybody in this panel has already spoken about it, where we are actually generating different kinds of data sets using single-cell multi-omics technology. This builds a huge information complex for which we will be needing a lot of advanced, tools and methods to actually, ingest this data, understand the complexities, the relationships, and then derive biological insights so that we can actually understand how the disease trajectory happens from healthy to disease phenotype over the time, and then specifically what kind of cells are actually following that journey so that we can then intervene and intercept at that level, not only in the detection, but get better understanding of the molecular mechanism and improve the drug targets and therapeutics. So I want to finally end my slide is all these kind of different information that we can, accrue over molecular or spatiomolecular or histological and anatomical basically can help us to build an overall integrated multi-scale organ phenotype map. And this is applicable to not only one organ or tissue type, it can be scaled across different organs so that we can get a better understanding of the different cell types, their maps, how they are organized in time and space, the different epigenomic contributions, their cellular interactions, the tissue organization, which can then help us to not understand disease or mechanisms, interactions, both at the molecular and the cellular and the spatial level, the pathology, and then derive, signatures and new biomarkers that can help us, get better with our therapeutics and eventually get better at drug discovery. So I stop my talk, and I'm happy to take questions.
Thank you for that. Thank you very much, Vivek. Very interesting talk. Particularly have an interest in sort of the multi-omics approach and then the single cell. I think it's a very sort of exciting area at the moment.